Hello, good morning, students. How are you? Hope you are enjoying at home. It's me, Dr. Tejendra Kumar, Assistant Professor of University of Delhi. Today, I'm gonna teach you a new topic that is linkage and gene maps. This is actually part one, and um, it is cover it is covering numerical questions solution. Okay, I I should have to clear that it is not describing theory part. It is just simplifying, and it is just describing that how would you can solve the numerical questions based on these problems. Okay, so this video is part of series genetics. Okay, have enjoy. This is about me. This is disclaimer. Now coming to the our main motto. Okay, so before the starting some solving the numerical questions, I have to uh, describe some terms, because these terms i will I will discuss in middle of the video, so you can get confused. So that's why I am describing here before. So first one is gene. Gene is what? Gene is the basic part of DNA, and it is made up of the DNA, and it is arranged on a chromosome. Okay, so it is a just simple sequence of the nucleotides, and it is also a genetic unit which carrying characters from one generation to the next generation, means from parents to their progeny. Next one term is gene map or distance. Actually, as I told you that genes are arranged on the chromosome. So that's why spatial arrangement of these genes, or we can say that physical appearance or physical distance between the genes of on the same chromosome or on the same chromatid, that is that's determination is called as gene mapping, or their distance is called as gene distance. Okay, suppose here, as I given you a diagram, here is the strand of DNA, and you can easily identify that there are two part, as I marked as encircled by the green color, and also cut mark with the red color. Okay, so between these two red and encircled with the green, this is suppose a gene 1 on the left side, and on the right side it is gene 2. Okay, so in between these two genes, one another sequence is also there, and that is the distance between the genes. Okay, so genes are actually arrangement on the chromosome, and they are sometimes closer to the each other, whereas sometimes they are more distant apart. Okay, now the next term, recombination. Recombination is what? Recombination is actually crossing over. Okay, crossing, and you know very well that crossing over occurs in the meiosis, and it is found in the germinal cells. Okay, or cells which forms gamete. It does not present in the somatic cells. Okay, so recombination is the actual it having exchange of genetic material between the different chromosome, and these different chromosome are homologous chromosome, and homologous chromosome are those chromosome which are part of a same pair. Okay, so it means they changing their genes between the same pair, and this shifting of gene is called as recombination or crossing over. Second one is linkage. Linkage is what? Linkage is just opposite to the recombination. As I told you that recombination is shifting or exchanging of the gene, whereas linkage, it opposite to the recombination, it opposes to this exchanging. Okay, so that is linkage. Actual linkage is closeness or close association of the genes on the same chromosome. I pictured here. You can see here in the diagram A, there are two chromosomes which uh, come closer and exchanging their genes in the B and C. It is after the dikinesis, whenever you find that both these parts get separated with the different gene, and this sometimes changing exchanging their some genetic material or some segment, that is that actual segment I marked here with the with the black circle and red and blue color. Okay, so these encircled region get exchanged. Okay, so this region is gene which is changing, and that's why it is called as crossed over here occur, and this portion get recombinant. Okay, and every gene have suppose this portion you identify and you check, then there may be many genes, as I mentioned here in the lower diagram, that there are sometimes genes are closer to the other genes, whereas sometimes their distance is more apart. Okay, some another terms are here: parental and recombinant. What is the parental? Parental sequence is the original sequence with from where you are starting the question. Okay, so starting sequence is called as parental sequence. Suppose you are working on an experiment and you find a sequence, then you say that okay, this is the parental sequence. Every sequence can be parental or every sequence can be progeny, or it's a progeny sequence. It's depend on you that which sequence you marked as parental, and after that all the sequence will be formed is called as progeny sequence or will be nascent sequence. They will not consider as the parental sequence. Okay, so parental sequence in any question, if you have given a problem or question in that, and there is saying that okay, this was the first sequence, and after that how many replications or how many crossing over it forms the another one, this sequence. So you have to just click on mind that first one sequence which you read in the question, you have to means first one from where all the sequences are originated, then that first one will be counted as parental sequence. Okay, and after that all the sequences which show the crossing over will be counted as recombinant sequences or recombinants. Recombinants are of two types: one is single crossover SCO and another one is double crossover DCO. Single crossover are those sequences which are generated by the single time exchanging of a gene, and double crossover, it means they exchange their genetic material but at 2 points. Okay, suppose there is a DNA sequence. i I took here three examples you can identify in these diagrams. First one is showing that there is a blue color uh, chromosome having three genes, 1 2 3, and below it there is a red color chromatid, and it also possesses 1 2 3, and after a generation this is showing same as it was, so it means there is no recombination. Okay, but in the case of second, number 2, here 1 and 2 number gene are exchanging. Okay, so that's why red color gene is shifting from lower to the upper side, and it also carries the 3, or you can say that 2 and 3 are were on the same side but one shifting, so it depends. Okay, but there is shifting of only one gene or only a single part. It means this is a single crossover. And in the situation third, you can see here that 1, 2, 3, there are 3 genes already available, but crossing over occur between 1 and 2, and again 2 and 3. It means there are 2 times they exchange their genetic material in between the homologous chromosome. Okay, so at the 2 sides, whenever they change, at the 2 points, whenever they change their genetic material, then it will be called as double crossover. It might be of 2 types. After the crossing over, you can see here that suppose it may be a red color shifted upside, or blue color shifted lower side, or it may be vice versa. But after all of this, you can find that if there are 3 genes in a sequential order, then after the double crossover, only a second number gene, or only a middle gene can be changed, and remaining both of they get their original position. Okay, means they get their same like structure. It is also can be explained by the another example, like there are suppose three children were sitting in a row, facing their mouth in the same side. Okay, but you change a number one child's face to the opposite side. Okay, it means now second and third are facing on the same side, but first one is opposing. Now you again shift any of one, means any of second or third on the opposite side. It means now what happened? It means first and second are facing same side, but third one is opposite. Or you also can say that first and third one facing on the same side, but second one is opposing. It means if you change the 2 times, it means uh, you change first and second, and then again shifting between the second and third, then it will change only in the middle, because you are shifting between the two, first and second, and then again second and third. Then after overall, second or middle one get their different position, but first and second will get the same, or otherwise we can say vice versa. If first and third get the opposite position, it means second will get the same. So overall, its conclusion is that only middle gene will be shifted. Some another terms are here. Coefficient of the coincidence, it means efficiency of 2 things happening together. So this is called as, that's why it is called as coincidence, means happening together. Okay, so simultaneously crossover of 2 or more genes is called as coefficient of coincidence, and it is also helpful for the measure the interference. Interference is what? Interference is simply meaning blocking. Okay, and it is just opposite to the coefficient of the coincidence, and it may be due to the linkage. Okay, if recombination at one site of chromosome, then it decreases the recombination at the nearby genes, means the decreases probability of the next one gene. It is called as interference, means they interfere, they blocking. Okay, some formula, because I already told you that today we will discuss about some numerical questions. So formula are most important. So what is the formula of the chain distance or recombination frequency? I mentioned here. Which thing is asking to you, you have to put on the upside and divide it by the total and multiply with the 100. You will find the overall result. So number of the offspring, because asking a question about the recombination frequency, then uh, uh, you will divide the recombination offsprings number by the total offspring and multiply with the 100. Okay, or in the another term you can say that you have to add all these single crossover and double crossover which are showing that particular sequential changes. Okay, I will discuss this in the next part. Next slide I'll mention there. Okay, so unit for this recombination frequency is 1 map unit. 1 map unit means 1% recombination can be occur there. Okay, it is also written as centimorgan, and centimorgan is coined the term to the honor scientist Morgan, T.S. Morgan, who discovered the linkage. So that's why it is given to the given honor to them. Formula for the coefficient of the coincidence, it is simple observed double crossover upon expected double crossover. It is sometimes similar looking like chi-square test, right? So but there is something different. Observed double crossover upon expected double crossover, or in the another term you can say that observed double crossover total. Suppose there are two double crossover, then you have to total all those things and divide by calculated uh, differences between the one set of gene and another set of gene. Okay, so both these differences which you have calculated, multiply them and again multiply with the total and then divide by this value to the double crossover summation. So it will give you the coefficient of the coincidence. Interference, it is very similar, as I told you that it is just opposite to the coefficient of coincidence. That's why you can simply it's solved by the 1 minus coefficient of the coincidence. So how to solve the numerical question? It is our main purpose. Coming to the one example, I took an example. Here you can see that I mentioned here how and what you have to identify. So there are some questions. First one, you have to identify the gene order, then determine the distance between the genes XZ, XW and ZW. Okay, and values are given in the table which is showing right side here. And then after the determining the differences, you have to draw a genetic map or gene map, means physical appearance, and then calculate the interference or coefficient of the coincidence. Okay, so values are given here. So how will you calculate? Okay, come to the solution. I divided this solution into some step by step parts, so that's why I mentioned here steps, so that you do not get confused. It is very simple. Step 1, identify the parental single crossover and double crossover. So how could you identify? First of all, parental. Overall values which given to you, out of these you have to identify two two sequences which showing the highest large number. Okay, so this is the parental. Simple, very simple. Okay, next one is double crossover. How could you identify? Just opposite to the parental, means you have to find out two least number sequences. Okay, means in this table 230 and 240 are the highest number, that's why it is two sequences are parental, and 12 and 8 are the least number in this table, so that's why these two are double crossover. And rest of all things, there may be four sequences. Okay, so rest all four sequences showing the single crossover. Okay, so there are total 8 sequences. Now you can identify that, okay, yeah, there are two parental, means highest two 230 and 240, and lowest two are double crossover, means 8 and 12, and rest all, means middle all sequences are single crossover, means 96, 104, 138 and 142. These are actually phenotypic ratios and phenotypic number, but actual sequence are like X, Z, W or etc. Okay, step second, identify the gene order. It is also very simple. And how could you identify? You have to compare parental and double crossover sequences. So I mentioned here parental with the red P color, so you should not get confused, small x and capital X and like so on. So that's why I changed the font style, so you can easily understand. Okay, so in the left side you can see that capital X, capital Z, capital W, these are parental, whereas small x, small z and small w, this is the another parent. Okay, you already know that in the genetics there are two alleles for the every gene, like capital T, small t, so that's why these are same sequences. Next one is DCO, double crossover. Double crossover is showing that small x, capital Z, capital W, and capital X, small z, small w. It means which allele or which gene get changed. You can easily say that, okay, in the parent and in the double crossover, only X gene has changed. So I mentioned here, find the odd one. Which is the odd one? X is odd one, because it is get changed from the parent to the double crossover. And I already told you in the previous slide that double crossover position will be changed and remaining two will be same in the middle one will be changed and remaining two will be the same in the double crossover, right? It means who one is changing, place it into the middle and redraw the sequence, and that will be the ZXW. So it's your exact gene order. So what is the exact gene order? ZXW for this given table. Okay, now coming to the step third. Step third is to determine the distance between the XZ. Okay, then again determine the distance between the XW, and finally we will again solve that what is the difference between the Z and W. Okay, so first one is XZ. So as I told you that there are some sequences which are parent and some another sequences which are double crossover, right? So I'm again coming back here. Parental, this is the capital X, capital Z and capital W, whereas double crossover, this is not showing like. And one another parent is showing small x, small z, small w. It means a single parent showing the same type of sequence, means capital letter or small letter. Okay, so we can say that we have to just identify the odd one. Means if there is capital X and capital Z there, we should not calculate them. Okay, but if there is capital X and small z, we have to calculate them here. So they will give us the distance, that how the X and Z get separated and change their sequence. Okay, and vice versa, small x and capital Z, right? So here you have to total the all these sequences which are showing difference different, like small x capital Z and capital Z small x. Okay, and add all these sequences, divide with the total and multiply with the 100, and you will get the result like this. So capital X and capital Z, where it is found, you have to identify in this sequence, in this table, and there is number 8. There is capital X, small z, small w, right? It means capital X and small z is there. Another one is 104. You can easily see here that capital X, small z, and capital W. We have to just only focus on the X and Z, not on the W, right? Because we are determining the X Z difference between the X and Z. Okay, so what's going with the W? It's not our concern. Okay, so all sum these all numbers and divide by the total is the 970. You can calculate 230 plus 240 plus 212 plus 8 plus 96 plus 104 plus 138 plus 142. Okay, so these all give you total 970. And after this calculation, you have to multiply with the 100 for the calculate in the percentage type value. Okay, so xz is showing the 22.68 mu. Actually it is written as mu or map unit, or it is also mentioned as the centimorgan, which I discussed in the previous slide. Now step 4 is for the determining the distance between the X and W. So how would you identify the X and W? X and W, it means you have to again find the odd one, means capital X and small w, and small x and capital W. Add all these numbers and divide by the total and multiply with the 100. You will find that 30.93 mu. Okay, so these are very simple. One more thing I want to clear here, that if there were parent is different, suppose in the like this previous slide, suppose this one parent was capital X, small z, and capital W, and another parent was small x, capital Z, and small w, then what would you calculate, or what would you concern in this next calculation? So that time you have to count that capital X and capital Z are where. That time you did not count, that time you should not count that capital X and small z, because it means they were similar to the parent, and you should not calculate parental sequence. You are determining the recombinant, so that's why you have to identify that odd one which are not present in the parent. Not it doesn't mean that you have to identify on the basis of the capital letter and small letter, right? Okay, so capital letter and small letter is not a criteria. Our criteria is that we have to identify the parental sequence exact which is present in the table. Okay, so what situation is given in the question, you have to focus on that. Okay, now again coming to the last distance, that is between the Z and W. This is again same process, but here is something different. Why? You can see formula here. Here I changed SCO and DCO differently. So why I changed? Because as I told you that double crossover is showing two time changes, whereas single crossover changing only one time. It means whenever you are uh, taking a number of the sequence for the double crossover, then you have to multiply with the two. Otherwise, it will give you false result. Okay, so for previous for previous slide, why we not uh, uh, taken as double like 12 8? Why? Because that answer is gene order. Coming to the again previous slide, here you can see that gene order was Z, X and W, not X, Z and W, right? So Z, X and W, it means who are on the edges, who are on the periphery, Z and W, right? So whenever you are calculating the distance between those two genes which are present on the edge or which are present on the just periphery, then you that time you have to calculate as doubled sequence for those which are showing the double crossover, right? Otherwise you should not multiply for the single crossover. Okay, and that xz and this is in this step 3 and step 4, xz and xw were not double crossover, were not showing the double crossover. That's why we calculated as single time, right? Okay, so the final calculation is sometimes not exactly different, just similar, but one small changes is that you have to twice the number of double crossover only. Okay, so you can check here 104, 142, 96 plus 138. These are all single crossover, while 12 plus 8, these are double crossover, and that's why we multiply with 2 here. And finally divide by the 970, and you will um, divide by 970 and multiply with the 100, you will find the result, and that is 53.61. One more thing I want to again clear, that if you are identifying the distance between the two genes which are separately uh, and which are present on the first and third position, not first and second or second and third, means they are distant apart and they are on the periphery or they are on the edge of that sequence, then you have to calculate all those data, and all those data you have to add except parental, because I told you again and again that we are calculating recombinant frequency, not calculating parental. Okay, so that's why I calculated here all these data like 104, 142, 96 plus 133, oh sorry, 138, and again added number of the double crossover also 12 and 8 and multiplied with 2. Okay, then divide by the 970 and multiplied with 100, and we will find result, and this result is actually in 53.61. Now what was the value between the xz? This is the xz, and xz is showing 22.68, whereas another next one is xz. Oh sorry, I mentioned here mistakenly xz again in the step 4. You have to correct it as xw, right? So xw in the red color, xw distance is 30.93, and last one here, zw is 53.61, right? So in the next step we have to draw map here. This map is showing 22.68, the difference 30.93 and 53.61. Sorry, I forgot here to mention the x, z, x and w here. So you can draw whenever you can draw in your notebook, then mark here at first position a first red line which is showing before the 22.68. You have to mark here x, sorry, z, and after the 22.68 red line, here you have to mention uh, x according to the gene order, right? And and last on the last end after the 30.93 you have to mention here w. So it is the exact gene sequence, and you can add and recheck here that 22.68 plus 30.93, overall it gives you value 53.61, right? So this is the uh, exact gene map, and total value is our total value is same. It means our calculation is perfect. Next solution is a coefficient of the coincidence, step 7. Okay, so for the coefficient of the coincidence you have to calculate double crossover and sum all these double crossover and divide by the distance between the zx and xw, as I told you in the previous slide that you have to take calculated distances. Okay, means observed upon calculated into 100 should be divided 100 should take as denominator, not on the upper side, right? So 20 divided by the 20, from where it get 12 plus 8, because there are two double crossover 12 and 8, so that's why I added both these are giving 20, and again I multiplied here these values 20.68 and 30.93, and again multiplied with the 970, then it gives me the value as 67.997. Okay, and after the division it gives me the value 0.297, and that is the exact coefficient of the coincidence. Okay, and last one is interference. Interference is very easy and simple. You can just minus the coefficient of coincidence value from the 1, and this will give you result 1 minus 0.297, and exactly it will be give you about 0.7. So interference is 0.7. Okay, so it means we can say that overall conclusion is here that interference is high, means 0.7 on the basis of 1, right? And coefficient of coincidence is about 0.3, right? So it means recombination can occur very low at in this progeny, or in this phylogeny, which is given here in this phenotype. It means their genes are more closer, that they rarely divides, or rarely show separation, rarely show crossing over, and these are linked more the time. Okay, so this is lecture, and thank you for your kind attention. Hope you will enjoy. Good day.